Today I will be discussing about mitochondria. Mitochondria may be defined as an organelles which are present in all the eukaryotic cells, and their main function is to produce ATP. That is why they are also called as energy transducers or powerhouse of the cell. Besides other functions. So let us start with an overview of mitochondria. These mitochondria are considered as unique organelles along with chloroplast. Why? Because they have their own DNA and they have their own ribosomes as well, because of which they are able to make their own proteins and ribosomal RNAs and transfer RNAs. And even though it is not like it is completely autonomous, it is still dependent on nucleus for majority of its proteins. But owing to having their own DNA and ribosomes, they are considered as semi-autonomous organelles. Now, history, the mitochondria were first discovered by Colliker in the flight muscle cells of insects, and these were named as phyla by Fleming at that time, and later they were renamed as bioblast by Altman, and finally by Benda was the one who gave the term mitochondria to these organelles, who coined the term mitochondria. Shape, size, number, shape varies. The shape of mitochondria varies. It may be highly convoluted, or it may be sausage in shape, or it may be disc shape, or it may be oval in shape. And mostly we make it oval in shape in eukaryotic cell for the representation purposes. And their number again varies a lot. It can be as low as 1 in Chlamydomonas or in Chlorella, and its number may go up to 100 or even thousands in the cells which are under intense metabolic which are in intense metabolic state. For example, in muscle cells there is a constant demand of ATP. Are in the liver cells or in kidney cells, even in brain cells which continuously needs ATP energy. So these cells have abundant mitochondria, plenty of mitochondria to keep up the energy demands of the cells. Next is the structure of mitochondria. So as you can see here in this is a three-dimensional structure of mitochondria showing on envelope. This envelope is made up of two membranes, outer membrane and inner membrane. In between there is a space which is called as intermembrane space. So you can say the envelope of mitochondria is made up of two membrane, the outer membrane and the inner membrane, and in between there is an intermembrane space. Chemically, the outer membrane, it is just a phospholipid bilayer, and in this phospholipid bilayer there is roughly equal ratio of lipid and proteins, and it is highly permeable. It allows the passage of ions and small molecules of approximately 6 kilodalton, which are less than 6 kilodalton, 6000 daltons. They allow free passage of these substances across the surface, across the outer membrane, owing to the presence of porins, large protein channels. They allow free passage of these substances. However, the inner membrane is selectively permeable. It restricts the entry of most of the substances and allow only a limited set of substances to enter or leave the mitochondria. This inner membrane, if we talk about the chemical composition of this inner membrane, it is more, protein is more, 3 times more as compared to the lipid component, and it is highly selective in nature. This inner membrane, as I said earlier, it is thrown into the, there are invaginations. This inner membrane frequently invaginates to form a structure which is called as crista, and in plural it is called as cristae. So you can see these cristae are in there, and why these cristae are there? So as to increase the surface area of the mitochondria or the inner membrane. As this uh, Besides Christie, you can see this pink background over here. This is called, inside this ground substance is known as matrix. And this matrix contains a lots of enzymes. For example, 50% of the proteins in this mitochondrial matrix is in soluble form. uh, and a lot of enzymes are in there which are involved with the Krebs cycle or the beta oxidation of fatty acid or amino acid metabolism or the urea cycle. So lots of enzymes are in there. And you can see this circular DNA is in there. As I said, that mitochondria contains its own DNA, so there are copies of this mitochondrial DNA, small circular DNA molecules in there. So besides this DNA, they have got their own ribosomes too in here. So this is all about the structure of mitochondria. So what did we study in this? What did we study in this structure? So that this it is outlined by two membranes, outer membrane and inner membrane, and in between there is a space enclosed which is called as intermembrane space. Then again this inner membrane is invaginated to form cristae so as to increase the surface area. And inside the mitochondria there is a ground substance which is called as matrix, which accommodates a lots of enzymes and proteins to carry out the metabolic processes which mitochondria does. So, after the structure, next is the origin, how this mitochondria originated in eukaryotes. So, there is an interesting theory behind this, which is called as endosymbiont theory, put forth by this Altman. So, it says that long ago there was a host, and this host accidentally engulfed a prokaryote or a bacteria, aerobic bacteria, and then later they both this bacteria stayed inside the host and continued in there as in and entered into symbiotic relationship with the same, because prokaryote was getting the shelter and protection inside the host, and the host was gradually became dependent on that prokaryote for the energy requirements. So regarding the host, what it was actually, it a prokaryote or it was a eukaryote or it was an archaebacteria, there is still mixed opinion about it, and still we are not very sure what it was. But as we know of today is that it was eukaryotes contains mitochondria, which is still believed to be a remnant of that prokaryote which it accidentally engulfed a long ago. So after this structure of mitochondria, let us move on to the so here are the brief functions of mitochondria. Besides ATP generation, mitochondria is involved in these many functions. So let us start with fatty acid metabolism. So fatty acids enters the mitochondria, and here they are broken down into acetyl coenzyme A via a process called as beta oxidation of fatty acid. And these molecules of acetyl coenzyme A are further fed into Krebs cycle, and it is further used in the production of ATP. In amino acid metabolism, the surplus amino acid also enters the mitochondria, and here they are deaminated. And after deamination, the ammonia is converted into urea through this urea cycle. Part of urea cycle occurs in the mitochondrial matrix, and a few steps are carried out in the cytoplasm. So after that, gluconeogenesis. It is the synthesis of glucose from the precursors like fatty acid or the amino acids, glucogenic amino acid. Glucose is synthesized in the mitochondria. Calcium balance. How calcium balance? This mitochondria it acts as a reservoir of calcium ions along with endoplasmic reticulum, and it helps in maintaining the calcium ion concentration in the cytoplasm. And protein transport. So as we have already discussed that 95% of the proteins of the mitochondria come from they are encoded by the nuclear genes. So when these proteins are synthesized on the cytosolic ribosome, they enter the mitochondria by crossing this double bilayer barrier, and after entering the mitochondrial matrix with the help of chevrons, they are folded and trimmed with the help of proteases and rendered functional, and they perform whatsoever their function is. So they are also concerned with the protein transport. the proteins which are destined for mitochondria. Besides, it is also involved in apoptosis. Apoptosis is programmed cell death. The cells which have completed their lifespan or the cells which become infected or cancerous, they enter apoptosis. And in apoptosis, mitochondria plays a key role. How? It releases a cytochrome C. It's a protein which is released in the response of the stress signal. And once the cytochrome C enters the cytoplasm, it activates the caspases, which brings about the apoptosis, which leads to the death of the cell. Okay, besides this function, let us discuss the main function of mitochondria, which is ATP production. Okay, ATP in this, first of all, glucose enters the cell. In the cytoplasm, glucose is broken down into pyruvate, and this process is called as glycolysis. In the second step, this pyruvate enters into the mitochondrial matrix by crossing this barrier, and here it is converted into acetyl coenzyme A. This acetyl coenzyme A is further fed into the Krebs cycle or TCA cycle, and it is converted into CO2 and water. So two steps are done. So in second step, NADH and FADH2 is produced. Now, the third step is electron transport and oxidative phosphorylation. Now, what happens in this step? The reducing equivalents which were generated during the Krebs cycle. What are reducing equivalent? NADH and FADH2. These molecules which were generated during the Krebs cycle and during the glycolysis in the cytoplasm, these are now oxidized at two complexes, complex 1 and complex 2. NADH is oxidized at complex 1 and FADH2 is oxidized at complex 2. Now, what happens when this NADH is converted into NAD plus? Hydrogen is released, which further break down into proton and electron. And this electron is picked up by this complex 1 and it moves downhill in an energy dependent process, meaning from higher energy carrier to lower energy carrier it is transferred. And while it is transferred from complex 1 to complex 3. Okay, so when it is moving downhill, energy is released, and this energy is used in the transportation of proton from this matrix to the intermembrane space. So, the electron move from complex 1 to complex 3. Now, when it is passing down the complex 3, again energy is released and the protons are moved from the matrix to intermembrane space. And when it moves through the complex 4, again energy is released and the protons are moved from matrix to intermembrane space. So finally, the electron is accepted by its terminal acceptor, which is oxygen, and it joins with proton and water molecule is formed. So as you can see, when a molecule of NADH is oxidized, a total of 4 plus 4 plus 2, 10 protons are transported from the matrix side to the intermembrane space. While when although it is not shown here, but here in complex 2, FADH2 is oxidized to a molecule of FAD. So you can see only 4 plus 2, 6 protons are released from the matrix to intermembrane space. So now what happens next? Now it is oxidative phosphorylation. So what happens in oxidative phosphorylation? Now as you can see, as the electrons are moving down this over here in this electron transport chain, a lot of protons are being transported from matrix to the intermembrane space. So slowly a gradient is built up, an electrochemical gradient is built up over here. So eventually the proton concentration becomes high in intermembrane space as compared to the matrix side. So of course they will move back down the concentration gradient, but they cannot move through these complexes. Now they can fall back into the matrix down the concentration gradient only through this complex. What is this? This is complex 5, sometimes also known by the name of F0 F1 particle or elementary particle or oxysome or ATPases. So as the proton, okay, let us see the structure first. So it is made up of two parts, F0 and F1. In F0 already there is a channel. So when proton move down the concentration gradient, like from intermembrane space to matrix, when they move through this channel, this part F1, it undergoes a conformational change, because of which here already ADP and inorganic phosphate are in there, which gets attached and ATP is synthesized. So when proton move from the region of higher concentration to a region of lower concentration, energy is released, a lot of energy is released, which changes the conformation of this F1 part, which brings about a rotational catalysis and helps in the formation of ATP. So when four protons moves from this side to this side, one molecule of ATP is released. Now let us look at the energy yield or energy of stoichiometry. So you can see in glycolysis and during Krebs cycle and during oxidative phosphorylation, 30 or 32 ATP are generated. It depends on how this pyruvate goes into the mitochondrial matrix, whether 30 or 32 ATP would be released. So this is all about the structure and functions of mitochondria.